Hi, welcome back to anatomy classes. This is Dr. Rajitha and today we are going to continue with the revision of systemic histology. So that is the last part of histology slides where we are going to revise in systemic histology. And if you like my work, don't forget to subscribe, like, share and click on the bell icon to get more updates about further notifications. So here is the systemic histology part 4, and if you are interested to know about the systemic histology of part 1, part 2, part 3 and general histology part 1 and part 2, I am posting them in my description box. Please check it out, and we shall continue with today's histology slides. So the slides here are, today we are going to do with the nervous system, that is cerebrum, cerebellum, spinal cord, and special senses, cornea, retina, and the last bonus slide is the placenta. So here is the first slide, which is called as the cerebrum, which forms the major part of central nervous system, and cerebrum is identified by the outer grey matter and central core is formed by the white matter. So the grey matter is also called as cortex and the white matter is called as the medulla. And if you see the grey matter, they are like laminated, they are present in layers. So there are 6 laminae, 6 layered structure it is, and it is difficult to differentiate each laminae at 10x magnification, but still we can see few layers. And the topmost layer is the molecular layers. Fusiform cells, neurons are also appreciated, which are called as Martinoti cells. So these are the different layers of cortex, and the cells vary based upon the what type of cortex it is. If it is a sensory cortex, we see more of stellate cells, and if it is a motor kind of cortex, we will find more of bed cells or pyramidal cells are more prominent. So here at a higher magnification, that is at a 45x magnification, where we are seeing the large pyramidal cells which are called as bed cells. And if you see these pyramidal cells, the shape, it is like this, like a triangle, like in cut section. And we are seeing apex of the pyramid is towards the cortex, outer surface of the cortex, and base of the pyramid is towards the core, that is the medulla or the white matter, and the axon arises from the base of the pyramid, and each angle we can see some dendrites arising from each angle of the pyramid. So this is how the pyramidal cells are present in the cortex. Let's see the layers. The outer layer is called as the molecular layer or plexiform layer, where more of nerve fibers are there and few horizontal cells. And external granular, granular means remember stellate. So they are more of star-like cells, stellate cells. And external pyramidal, again small sized pyramidal, small and medium sized pyramidal cells, and internal granular, again stellate, and they have a band of white matter within the internal granular, which is called as external band of bilarger. And internal pyramidal granular ganglionic cells are called as large pyramidal cells of beds, and this layer also consists of a band of white matter within it, which is called as internal band of bilarger. And the last layer is the multiform or pleomorphic, where we find different varieties of cells. So the next slide here is the cerebellum. The cerebellum is also part of the brain. So we find the outer grey matter and inner core is formed by the white matter, and grey matter is called as the cortex. And here the grey matter is present in 3 laminae, that is 3 layers. So we will see one by one here. So the core, here is the core, I am drawn a line in the core, that is formed by the white matter, which is formed by the axons arising from the cells, and the grey matter is present in 3 layers. So the whole thing what we see from one end to the other end is called the folium of cerebellum. The cerebellum is identified by the folium, where the white matter divides like a branching of a tree, which is called as arbor vitae cerebelli. So this is the whole folium from one end to the other end, and from here we can see the other folium from the other side, and the core is formed by the white matter, and grey matter, the outer from here to here, which is little paler in color, the outer part of the cortex is this, This is called as the molecular layer. So molecular layer consists of mainly some basket cells and mainly for nerve fibers, that is the molecular. And the second layer is here where we see a single line of cells. So it is a single celled layer identified by such a large cells. They are flask like cells. I am going to show in a higher magnification too. These are called as Purkinje cells. So by seeing the Purkinje cells you can tell it is a cerebellum. So Purkinje cells are flask like where the base of the flask is towards the white matter and the tip of the flask is towards the outer cortex. And inner third layer is identified by dark pigments with so many dots because it is having so many cells which are called as granular cells, and so it is a granular layer. So the principal cells of the cerebellum are the granular cells, which are the only cells are excitatory in function. Rest of all the cells what we see, the Purkinje cells, basket like cells, horizontal cells, all these are inhibitory in function. So the chief cells of cerebellum are the granular cells. They are more prominent in the third layer. And the core is formed by the white matter. So here is the core of the white matter. So let's see at a higher magnification. So we can see here this is the molecular layer from here to here where it is slightly paler, and we can identify few cells in the molecular layer which are basket shaped cells. So these are the molecular layer, and we can see such a beautiful large cells are Purkinje cells, well easily identifying. And the base you can see here the nuclei of each Purkinje cell. So the axon runs down and they go into the white matter, and finally it anastomose ends by synapsing with the grey matter, that is the nuclei of white present in the cerebellum. The nuclei are like dentate nucleus, emboliformis nucleus. So there are various nuclei present within the core of white matter. So these axons will finally end up by synapsing in the core of white matter, that is within the intracerebellar nuclei. And this is the granular layer. From here to here is the granular layer, which are identified by mainly granular cells and some stellate cells. And this is the core. From here to here is the core. So let's see the identification points. It has got three layers, that is the cortex has got the three layers. So the grey matter here, the outer molecular layer consists of stellate cells, basket cells and dendrites of Purkinje cells. Second layer is a single cell layer, which is a Purkinje cell layer, which is the main identification feature of a cerebellum. And the third inner granular layer mainly consists of granular cells, Golgi cells and synaptic glomeruli, and medulla consists of white matter. The third part of the CNS, that is the spinal cord, I am going to discuss here. To see the spinal cord, no need to have a compound microscope. Only with a hand lens or a dissecting microscope we can identify it as spinal cord. And spinal cord, it is easily identifiable. We can see the outer coverings of the spinal cord here, the thin coverings we can see here. And the dura mater is not well appreciated in this slide, but otherwise the thick layer may be there outer to the spinal cord, that is called as dura mater, but which is not seen clearly. And we can see some blood vessels present in the subarachnoid space, which forms branches from the spinal arteries supplying the spinal cord. And it is identified by the posterior faint sulcus and anterior deep median fissure. We can see a deep median fissure on the anterior side, and in the center it is identified by the central canal, and it has got the central grey matter and outer white matter, which is the reverse of cerebrum and cerebellum. Here the outer is the white matter, central core is formed by the grey matter. So I am drawing an outline of a grey matter. We can see here this is the grey matter present, having an anterior horns and the posterior horns, and the posterior horns are narrow and the anterior horns are quite broader. And here is the central canal, and this is the anterior horn, which consists of mainly motor neurons, and this is the posterior horn, contains mainly the sensory neurons. And if the section is taken at the thoracic segment, we may appreciate the lateral horn also, but here it is not taken at the thoracic, so no lateral horn is there. And white matter, because of this gray matter, we are dividing into posterior column. So this is the posterior column, this is the lateral column, and here is the anterior column. So these are the differentiating features to identify it as a spinal cord. And here at a little higher magnification to see the central canal, well differentiated central canal is there, and we can identify a line separating the, here is the white matter, this is the white matter, and this is the gray matter, which consists of nuclei, motor nuclei, sensory nuclei, all preganglionic nuclei of sympathetic and parasympathetic, all these are present inside the gray matter. So identification points, central head-shaped grey matter with central canal, and white matter is having a deep median fissure and posterior median sulcus. The cornea. So the cornea, it is easy to identify by its the central core. Here we can see the uniformly arranged collagen fibers. The cornea consists of 5 layers. Each layer I am going to discuss now. And the cornea is a transparent membrane, makes the outer part of the eyeball, that is the anterior 1/6 of the eyeball where the iris is present. The black colored part is covered by the cornea, and rest of the white part of the eyeball is called as the sclera, which is a thick collagen fiber layer. So this cornea, it is transparent because the light should enter inside the eye through the cornea, and it has got the outermost epithelium, which is the stratified squamous non-keratinized epithelium. So this is the stratified squamous non-keratinized epithelium, and below the epithelial cells are resting over a basement membrane. So I am drawing a line over a basement membrane, and this basement membrane is called as anterior limiting membrane, and this also called as Bowman's membrane. This is called as a Bowman's membrane. Deep to the Bowman's membrane, we have got uniformly arranged collagen fibers, which is called as substantia propria or the stroma of cornea. And here the collagen fibers are arranged so uniformly, they are so arranged uniformly so that the light traverses through the collagen fibers easily. And we can see few cells, few nuclei we can appreciate between the collagen fibers. These are the fibroblast which produces these collagen fibers. And if we observe in the cornea, there are no blood vessels in the cornea. So it is an avascular tissue, doesn't have any blood supply. And deep to this, again there is a layer of a basement membrane which is called as inner limiting membrane, which is also called as Descemet's membrane. So, and D comes later, Descemet's membrane, over which there is a thin layer of squamous cells or endothelial cells. So the thin layer of squamous cells are present with a flat nuclei. So these are the 5 layers of cornea. So the outermost is the epithelium, then a basement membrane, that is the outer limiting membrane, then substantia propria identified by collagen fibers and fibroblast and no blood vessels, and deepermost is the again inner limiting membrane or Descemet's membrane and a layer of squamous cells. So here we can easily identify the outer stratified squamous non-keratinized epithelium. I am drawing a line over the outer limit, and we can see the nuclei here, nuclei of columnar cells, nuclei of cuboidal cells, and these flat cells are the nuclei of squamous cells. So it is stratified squamous non-keratinized epithelium. And we can even appreciate some fibroblast cells and collagen. Bundles of collagen fibers are there. We can see bundles of collagen fibers. So 5 layer outer stratified squamous non-keratinized Bowman's membrane, anterior limiting membrane is present, substantia propria identified by uniformly arranged collagen fibers with some fibroblast, and Descemet's membrane over which simple squamous cells are resting. So the retina is identified by so many layers here. So the retina consists of laminae. So there are 10 layers present in the retina. With 10x magnification, you can identify it easily because it has got well differentiated layers. So we can discuss all the layers in the higher magnification, but at 10x magnification, what we can identify is where I am putting an arrow, it is the layer of rods and cones, and this part where the dark part, those are the nuclei of rods and cones. Plexiform layers and inner nuclear layers, ganglionic cell layers are also present. Identify each layer. So the each layer, the outermost layer is the pigment cell layer, which are simple cuboidal cells containing melanin pigment and helps in the absorption of excess light. And next to the pigment cell layer is the rods and cones. So I am putting an arrow over the rods and cones layer, and deep to the rods and cones layer there is a line here. This is called as external limiting membrane. This external limiting membrane is formed by the processes neurons fibers from the Muller cells. And deep to the rods and cones and Muller external limiting, this nuclei, they are the nuclei of rods and cones, which are called as external nuclear layer. Deep to external nuclear layer is the, or outer nuclear layer is the outer plexiform layer. So there are plexus of nerves which are called as outer plexiform layer, and deep to which is a inner nuclear layer. So here is the inner nuclear layer. Inner nuclear layer consists of mainly bipolar neurons, nuclei of bipolar neurons and amacrine neurons, and these bipolar neurons will form the first order neurons in the retina. And next to the inner nuclear, then again inner plexiform, this is inner plexiform layers, and then ganglionic cell layer. This layer we can identify with large ganglionic cells. These are called as ganglionic cell layer, which forms the second order neuron in the retina. Next to the ganglionic layer, we have got the nerve fiber layer. All the nerve fibers compactly come together to form the optic nerve. And blood supply of retina, it is supplied by the ophthalmic, that is the central retinal branch of ophthalmic artery, and the outer part of the retina is supplied by the choroid plexus. So the layers, pigment cell layer, first layer, which is a cuboidal cell layer having melanin pigment and helps in the absorption of light. Rods and cones, which are main cells for the perception of colors. Both in cones are for bright light vision and rods are for dark light vision. And external limiting membrane form the nerve processes arising from the Muller cells. Then outer nuclear layer, the nuclei of rods and cones will form the outer nuclear. Then outer plexiform layer, nerve plexus of these rods and cones will form outer plexiform. Inner nuclear consists of mainly bipolar neurons, which forms the first order neurons. Then ganglion, inner plexiform layer will form the nerve plexus of all these bipolar cells and amacrine cells. And then ganglionic cell layer consists of large ganglion cells, and then the nerve fibers of these ganglion, which forms the second order neurons, they all compactly gets arranged to form the optic nerve, and the last is the inner limiting membrane. So these are the layers of retina. So let's look upon the bonus slide today, that is the placenta. Placenta, this is a 10x magnification. So under 10x magnification, you can identify the cut sections of chorionic villi. So what we are seeing is the cut sections of chorionic villi are present inside the placenta. And between the villi, the gaps between the villi, what we are seeing, there we can find some dots. They are the blood cells. They are the blood cells, WBCs, RBCs present, which are intervillar spaces present between the chorionic villi, and this intervillar spaces is filled with the maternal blood. And fetal chorionic villi, if we try to see in each inside the chorionic villi, we can identify the cut sections of blood vessels very nicely present here. So we can identify the cut sections of blood vessels. By seeing the blood vessels, we can see it as a tertiary villi. and these blood vessels are lined by the simple squamous cells which are called as endothelium resting on a basement membrane. And between these blood vessels, the tissue which is present, the connective tissue which is present, which is called as mesenchyme, it is derived from extraembryonic mesoderm. And outer to each chorionic villi, it is lined by cuboidal cells, and these cuboidal cells are called as cytotrophoblast. And outer to that cytotrophoblast, there are fused cells with where only the nuclei are present, which is called as syncytiotrophoblast. So the outer to each villi is the syncytiotrophoblast. So what is placental barrier? We know the blood is, fetal blood is inside the blood vessel. So here is the fetal blood, and maternal blood is present here outside in the maternal blood. So the layers between the fetal blood and the maternal blood are called as placental barrier, which is formed by the lining of the capillaries, that is the endothelium with the basement membrane, then extraembryonic connective tissue, extraembryonic mesoderm, which is forming the mesenchyme, then cytotrophoblast, syncytiotrophoblast. All these layers together called as placental barrier. And the placenta, when it is at term, the cytotrophoblast will get depleted, it gets reduced, and only syncytiotrophoblast is present to reduce the layers and increase the exchange of blood for demand of more nutrition for the developing embryo. So this is at a higher magnification where we can see each villi here, and inside the villi we can see some cut sections of blood vessels easily to identify, and the mesenchyme is here, and the cytotrophoblast is lining each one and surrounded by the syncytiotrophoblast, and we can see some blood cells also in the intervillar space which is filled with the maternal blood. So identification features for the placenta: cut sections of chorionic villi are seen, and villi containing embryonic connective tissue in the core, which is derived from extraembryonic connective tissue, extraembryonic mesoderm, and containing fetal blood vessels, fetal capillaries surrounded by inner cytotrophoblast and outer syncytiotrophoblast. So thank you all for encouraging me and motivating me with your nice comments, and I'm happy to share that subscriptions crossed more than 2500, and if you haven't subscribed, please subscribe to my channel and click on that bell icon which is right next to it to get more updates about my future classes, and please mention the topics you need in my comment section. Bye, take care.